The longest part of the mitotic cell cycle is interphase, a phase when the cell grows and develops in preparation for mitosis. The DNA in the cell and many of the organelles replicates during this time period. The chromatin, which contain the cell's genetic information, are spread out inside the cell's nuclear envelope and become uncondensed. The cell also contains a pair of centrosomes, which are comprised of centrioles, and are the organizational sites for microtubules. Next is prophase. During prophase, the chromatin all condense while the nucleus disappears. The centrioles begin to form microtubules that extend from the centrioles to create a mitotic spindle. The centrosomes begin to move to opposite poles of the cell. Asters surround the centrosomes and are the thin lines extending from them. Sister chromatids appear and pair up and are identical to one another. Metaphase follows prophase. First, the nuclear envelope fragments or breaks down, allowing the microtubules to extend and invade the nuclear area. Next, kinetochores attach to the microtubules. These become kinetochore microtubules. Kinetochores are specialized protein structures located at the centromere. Other non-kinetochore microtubules work with those opposite from them. During metaphase, the longest stage of the cell cycle, the sister chromosomes line up at the middle of the cell, forming a metaphase plate, which is an imaginary line that runs across the middle of the cell. The centrosomes are now at the opposite ends of the cell. For each chromosome, a kinetochore is attached to each sister chromatid that runs back to a centrosome. The entire set of microtubules makes a spindle shape. Next is anaphase. In anaphase, the shortest stage, the chromosomes separate, yielding the sister chromatids each getting pulled toward a different pole, making each chromatid a true chromosome. An equal number of chromosomes are pulled towards each pole. Centromere first because the microtubule is attached to the centromere region. This movement causes the kinetochore microtubules to shorten and non-kinetochore microtubules to lengthen, elongating the cell. During telophase, the daughter nuclei form along with the nuclear envelope. The two daughter cells are genetically identical. Finally, cytokinesis occurs when the cytoplasm is split or divided between the two daughter cells. In plants, a cell plate is created to aid with the separation, and in animals, the cleavage furrow forms that pinches that cell in two.